Hello, Buntle. I hope you're good. So I'm gonna start with this is the first part, the first series of my life science videos. So I decided to make videos since we have this problem of load shedding. So I'm just gonna make these videos. So I'm gonna make videos for every topic basically for life science, just covering the basics so that you have you know material to go back to if like was stuck at any point. I wanna make this video so that you can watch them at your convenience. I want especially towards the exams. I wanna when you forgot something fella to have something to reference back to. So yeah, I hope you follow, and if you have any questions you just post it on WhatsApp, and then yeah, I will explain them um, in our one-on-one sessions. So we are gonna have basically less one-on-one sessions because I have these videos. So I'm gonna do these videos, and then after these videos, of course, with the questions that I give you every day. I give you questions every day, so you look at these videos and the questions I give you every day. If you have any questions there, like any at all, please post them go WhatsApp or write them down so that when we're gonna have our one-on-one sessions, um, we're gonna answer those questions and basically um, go through the process of how to attempt doing this in a test setting. One. So I would say the amount of work will increase. So the amount of work that we're gonna do right now is going to increase. However, um, we're gonna meet less often. So even though we're gonna meet less often, the amount of work we're gonna do is going to increase. So I'm gonna start here with life science. So with life science, I'm gonna start with life science. Particularly, I'm gonna start with paper one's work. Ne? So I'm gonna start with life science paper one, and there's basically 10 topics. Ne? Let me just go to the index and show you. So, okay, is it here? Yeah. So there are basically 10 topics, ne? And then six of them are paper one, and then four of them are paper two. Ne? So the one that I'm doing today, DNA, you already did, right? This one is paper two. So I'm gonna start with this one. So with life science, I'm not gonna do it like math, where I did like paper one first, and then I'm gonna do paper two. Because I get with math, we already done with all paper one content. I get so now with this one, I'm just gonna do basically from DNA, and I'm just gonna go down until human evolution with these videos. Ne? So I'm gonna post like videos here to life science more. And then I don't know, maybe when I reach somewhere here, maybe 6 or 7 or 8, yeah, somewhere here, then I think I'll switch it up and go to paper two, maths a bit, right? So that like rebel balance, and then come back here and then finish these ones. Because essentially I want us to cover like basically all of like your content, so that like um I'm sure like of your capabilities like in the exams that are gonna come. So let's start. So we're gonna do DNA. So the way I'm gonna do these topics, I'm not gonna go deep into them in a sense. Yeah, I won't go as deep as they go like like when they teach in school. Ne? I'm just gonna I'm just gonna cover basically the exam technique. So the I would say the level of understanding that you have to have for a test. One. Yeah. So let's start. The first one is DNA. DNA is paper two. Ne? So DNA is paper two, and yeah, that's the first topic. So it says life at a molecular, yeah, life at a molecular, cellular, and tissue level, right? So we're gonna start. So um Okay, introduction. So we know that all living organisms contain DNA, right? And and this thing called RNA. But the one that's most, I would say, most common, that the one that you commonly know is this one, DNA, right? And essentially, like, what is this DNA, and like, why do we do this topic? Because I believe it's very important, like, before you start with anything, né, like in terms of school wise, to know why you're doing it. So DNA, like, why are we required to know DNA? Why is it important? So with DNA, so just imagine all living organisms, right, have a basic cellular structure, right? So we have plants and animals, right? Both of them have cells, right? So all living organisms have cells, right? Either they are plant cells or animal cells, but they have cells, right? And then what happens during mitosis, right? You know, the process of mitosis, what happens is that these cells, they divide, right? And then an identical cell is basically produced, right? So how does um a cell like produce an identical copy of itself, ne? Number one. And how does, like, do you know, like, for instance, if let's say you get hurt, that wound will close up, and then your skin color, you will retain your skin color, right? Even when you cut your hair will grow back the same. You don't grow back pink all of a sudden, or brown all of a sudden. Like, as time goes on, yeah, maybe it will be white as time goes on. However, basically your structure, your cellular structure will be the same, right? That's because of one thing. That's because of this DNA, right? So it doesn't matter what part of your body, like, there is, like, it has um a part of your DNA, right? Because basically what happens is, let's say you cut your nails, your nails are still gonna grow the same way, right? Why? That's because that gene, like, there's a gene in that DNA, right? That codes for that, right? And I'll explain as time goes on. So basically, when you think of DNA is the coding, because that cells have different functions in the body, right? Because, like I said, you are made out of billions and of cells, right? Millions and billions of cells, right? So what happens is, these cells, like, all of them, right, they are made of this DNA. So there's some sort of instruction that they got, right? So that they know, like, their functions. Like, some cells are used for organs, some cells are used for the skin. Like, all of them, they have different functions and um basically properties, right? And this the DNA, basically, it's coding for all of that, right? So the DNA codes for all of that. So the DNA is just the code, right? It's not the structure itself, it's the code. But you'll see what I mean as I go on. So this is basically the cellular structure of a cell, right? I won't go through all of them, but what's important here is this nucleus. So the nucleus is where the DNA is contained, right? So it's contained in the nucleus, right? And then this is basically the structure of the nucleus. So what happens is that we have the nuclear pore, right? So this is where things can go in and out of the nucleus, right? And then also this membrane. So this membrane and this pore, like, they work together. However, you know that a membrane also, like, it's selective, right, in terms of what goes in and out, right? And then this is the nucleoplasm. So this is something similar to the cytoplasm, inside of the nucleus. So it's just a jelly-like fluid, right, to keep all of those organelles inside, right? Because you can't have life without this cytoplasm. Like, there must be some sort of fluid, some sort of liquid, right? If you can see, all living organisms have, like, water. So a sort of, like, liquidic jelly substance is needed for all of these organelles to stay suspended in, right? And then you have the nucleus, right? Here's the nucleus, and then inside the nucleus, right? So inside of this nucleoplasm, we have this thing called the chromatin network, right? Do you remember when I talked about the chromatin network when we started with meiosis, right? So this is the same chromatin network, right? And this chromatin network is the one that contains the DNA, right? So this chromatin network is the one that contains the DNA. Okay. So before before we move on, so what is like a DNA? So what is DNA really? I wanna, because we know its function, we know that it codes for something. So it's the one responsible for basically giving the initial instruction of how your body should be composed, right? Of how your body should look, of how your cells should function, right? So if that's the case, what is this DNA, right? So this DNA is a nucleic. So this DNA is a what? Is a nucleic acid. It's a type of nucleic acid. So this is very important. So DNA is a type of nucleic acid. Nucleic acid. So this thing here, nucleic acid. That's a DNA. So DNA is a type of nucleic acid. Now, before we continue, let's discuss what this is, right? So, what is a nucleic acid? A nucleic acid basically is a what? Is an organic compound. So, organic means that it's alive, right? So, it's an organic compound, right? So, it's an organic compound, right? So, a nucleic acid is an organic compound made out of what? Made out of a building block. So okay, at the term for building block in life science is monomer, ne? So monomer is building block. So this monomer term means building block. So what is a nucleic acid? Nucleic acid is an organic compound, right? Right, which so this nucleic acid is an organic compound. And how is it formed? It's formed by these nucleotides. So there's a compound, there's a building block called a nucleotide. Now we're gonna go to the structure of how a nucleotide looks and what is a nucleotide. But for now, just know that these nucleic acids, so number one, DNA is a type of nucleic acid. They are obviously there's more than one type. Is a type of nucleic acid. And what is a nucleic acid? A nucleic acid is an organic compound, right? Meaning it's alive, that is made out of nucleotides, right? So made out of monomers called nucleotides, or building blocks called nucleotides. So from now on, I'm gonna use the term monomer. It just means building block, né? So that like we get used to this terminology, right? Yeah. So that's basically a nucleic acid. So nucleic acid is made out of these nucleotides, right? And how many nucleic acids do we have? We have two types. We have DNA and RNA. So DNA and RNA are types of nucleic acids, right? Which are made out of building blocks or monomers called nucleotides, right? Now they said, okay. And what does DNA stand for? It stands for deoxy ribonucleic acid. Do you see? This nucleic acid is the deoxyribonucleic acid. You see it like it's already there in that it's contained even in the word of so the terminology of DNA. And then here we have RNA, which is ribonucleic acid. I I think these ones are I would say these ones are, I would say, are self-explanatory. You'll see this deoxyribonucleic acid, ribonucleic acid, that's where this R comes from and the D comes from, right? And then they said together these form the basis of all life on Earth, right? You see, they form the basis of all life on Earth. They don't really form all life on Earth, they form the basis of all life on Earth. So they are there. So the DNA is basically like I told you, it's there to give instructions. That you'll see the RNA is just the messenger of those instructions. The RNA is there to just transfer those instructions to other, like to you'll see when you do protein synthesis. right? But these two, basically, these nucleic acids are there basically to help with giving instructions to cells, right? And then this is the basic structure of a nucleotide, right? So what is a nucleotide? Nucleotide is composed of these three basically compounds, right? It's composed out of a phosphate group, right? So it's a phosphate group, a sugar molecule, right? And a nitrogenous base, right? So that's what forms that's the basic structure of a nucleotide. And these nucleotides, you find also inside here. So these nucleotides will also find inside the nucleus, right? As you can see, this nucleus, you see it's from the nucleus, right? It's from the term nucleus, which means you know where it's found. It's found inside the nucleus, right? And then, as you can see, this thing is held by bonds, right? These bonds are not really important, this one and this one, right? However, there is a bond that's important. I will tell you about that, but just know that these three are held in place also by bonds, right? And these bonds also have names, right? It's just that for your syllabus or for your level of study, you need to know the names of these bonds, right? You just need to know that a nucleotide, so the building block of a nucleic acid, which is nucleic acid, like I said, a nucleic acid is RNA or DNA, is what is a nucleotide. A nucleotide is made out of a phosphate group, a sugar molecule, and nitrogenous base. That's what you have to know, right? And this phosphate group is bonded to this nitrogenous. This phosphate group is bonded to this sugar molecule via this bond, and then this sugar molecule is bonded to this nitrogenous base via this bond, right? So that's the basic structure, basically, of a nucleotide, of yeah, nucleotide, right? So now that we know this basic structure of a nucleotide, what did we say? We said this nucleotide, right? What is it? It's a building block for what? For nucleic acid. So it's a building block for DNA and RNA. And let's just see, how does it form? So how do these building blocks form DNA and RNA, right? So, okay. Okay, let's move on. So, okay. Okay, DNA is found in two locations. Okay, for now, let's not do that. I want here the structure of DNA. So, do you see, this is basically the basic structure of DNA, right? So, like I said, we've got this, what is this phosphate group, the sugar group, which is bonded to the sugar group, right? And this is the nitrogenous base, right? This is also a bond, right? Do you see, this is also a bond, this is also a bond, but these bonds, they don't have names, right? For now, you don't need to know the name of this bond, right? And then what is this? Like I said, this is the nitrogenous base, right? Do you see how they bond? This is the nucleotide, right? This is one nucleotide. So let me just draw it here and illustrate it. So this is one nucleotide, right? Do you see it's bonded to another nucleotide here, right? Via this bond, right? It's bonded via this bond, and then, right, and then it forms this long chain, right? This long chain, right? Now, this chain, these nitrogenous bases, so the first thing it's going to form this chain, right? And then these nitrogenous bases, do you see them? These nitrogenous bases, right? What do? They have what we call complementary pairs, right? Complementary pairs. So complementary means that they have to be so like, for instance, adenine can only be bonded to thymine, you see. So that's what they mean when they say complementary. So it can only be bonded to a certain one. So A can only be bonded to T, as you can see, and G can only be bonded to C, right? So that's basically what does complementary mean. So you can even, if I can give you an example, you can even like look at it as like relationship. So this one, because like it's married to T, it cannot like be bonded to also G and C. Like it will never. A to T, G to C, just like that. So these bonds are complementary, right? Now, do you see that the way that, let's say, this left portion is arranged, it's going to affect the way that this right portion is arranged, right? Because the first thing, number one, that you have to know is that DNA is a double helix structure, right? So there's this side, right, of the helix. So I would say this line here and this part here, and then there's this side of the helix, right? So it's a double helix. And what is this bond here that these two nitrogenous bases are bonded to? This bond is called the hydrogen bond. So these ones are bonded via weak hydrogen bonds. Do you see? They are bonded via weak hydrogen bonds. And this is very important that you know that they are weak hydrogen bonds, because they can separate, right? Whereas these ones are not weak. These bonds are not weak. They are strong bonds, these ones. However, these ones are weak. So just know that they are able to separate. That's why the DNA, like you can create many sets of DNA via mitosis. So basically what will happen is that this will deattach, right? And then new nucleotides will form, and then you're going to have two DNAs, but we'll go to DNA replication when you are done. So just to sum it up, like I said, these are nucleotides, right? And then we have these nucleotides. What do they first? They are firstly bonded to each other, right? Via these strong bonds. This side, right? This one side, right? And then this other side will be bonded to these nitrogenous bases via these weak hydrogen bonds, right? So if I would explain this, the first DNA structure to basically, I would say, form in your body, right? It's made out of, obviously, a portion that comes from your mom and a portion that comes from your dad, right? They both combine, and then they will basically determine how will you look, like basically your genes or everything. This structure here is basically created by both parents here, right? Because your DNA has to have the genes basically of both your mother and your father, right? However, you'll see how this relates when we go to reproduction. But it's very important to see that this knowledge, like, it's not in isolation. Do you see that we start with DNA, and then as we go further, you see, you start with DNA, we go to meiosis, mitosis, and then we go on. So that you see that there's a link actually with this, right? So this DNA here structure, of course, it can replicate because of these weak hydrogen bonds, right? So after the mother and the father, basically, they have fertilized and implantation has happened, right? What will happen is the first stage of mitosis obviously will happen, right? Because it has to grow, right? That specific zygote, it has to grow, right? So what will happen is that there's going to be firstly DNA replication, and then from there on mitosis. However, I'll explain DNA replication further. So let's go. So like I said, these are the hydrogenous bases, right? And there are four types. There's adenine, cytosine, thymine, and yeah, this one. And then now you'll see that these complementary pairs, I get the form complementary pairs. These complementary pairs, they are simple. You see adenine to thymine, G to cytosine, right? This to cytosine. So just know these pairs. These pairs, this is not difficult, right? It's not difficult. You just have to know which one is paired with which for now, right? And then know that DNA is a basically double helix structure. Then from there on, we can move on. So like, okay, this I already explained that DNA really contains hereditary information, right? So genes in the form of genes, right? So hereditary means that you inherit, it comes from the word inheritance, right? So this information has been inherited like for ages. So the DNA that you have basically is I would say it's a complement, it's a yeah, compilation of all of that information, right? So it's a compilation of all of that hereditary information, right? Yeah, from your past ancestors to your grandmother, and then your parents, and then you, right? And then we that's basically the base. So we are done with the structure of DNA. So that's the structure of DNA, and obviously this is just a 2D representation of it. When they look at it through those microscopes, they see these double helix structure, right? So it's a double helix structure, right? And don't forget that it's a double helix. Why? Because it has these two sides. There's this one that formed originally, right? And this one forms on top of it, right? So this one and this one. And don't forget these ones, when they started this, so one from the mother, one um DNA basically comes from the father, right? And then, so I would say this DNA, the contribution of this DNA is done by both parents, but you'll see how it happens when you go to human reproduction or reproduction in general. So let's move on to RNA. So RNA, ribonucleic acid. So like DNA, RNA is also made out of these nucleotides. Why? Because RNA is also a nucleic acid, right? So now with RNA, there are many types of RNA, and the only really difference between RNA and DNA is number one, is the bases that are found in RNA and DNA, right? Do you see that in RNA we have this base like a uracil, right? So as you can see, it's not here, right? Here we have thymine in DNA, right? So RNA does not have thymine, right? Only DNA has that base thymine. So that's the first way you can see that this thing is RNA. And then secondly, RNA is only one strand. So you see DNA is a double helix strand, right? So RNA is only one strand. So it's not a double helix, it's only one strand. So that's the two differences between RNA and DNA, right? And then I would say the third difference is their function, right? The function of DNA is to send out information. So it's the command center, you'd say. It's the one that sends out the information. The RNA is the one that does the job, basically carries the information, and also yeah, translates the information, right? But you'll see that when you're doing protein synthesis, as you can see, there are three types of RNA, right? Messenger RNA, ribosomal RNA, and transfer RNA, right? So the RNA's function basically is to carry out the DNA's first instruction. So this one is basically the messenger guy, or I would say that if this is a business, these are the employees, right? That they actually carry out what the DNA, what the manager said like they should do. So these ones, RNA's function is to carry out what DNA. So DNA is the one who will give out the instruction, RNA will carry it out, right? So now we'll move, right? So where is the RNA found? So okay, before we go to these locations, I want to just show you the structure. So like I like I told you, the structure of RNA is a single stranded structure, right? Which is not coiled, right? And as you can see, this is a ribose sugar, right? And don't forget, here they used a deoxyribose sugar. Do not forget the difference. That's why here it's called DNA, there is called RNA. It's because of these sugars, right? There, it's a deoxyribose sugar. Here is a ribose sugar, right? Yeah. And then sugar molecule, yeah. And then okay, so as that's okay, this I already explained this, right? And then role of RNA. Okay, so the role of RNA is very important for what? For this, basically, for this protein synthesis. And that's where we started. Like that's why we actually looking at DNA. Why is this DNA important? Why? It's because of this protein synthesis, right? Because like I said, many things in our body, like okay, all things in our body obviously have cells, right? And most of our body is composed of this thing, protein, right? So protein has many functions in our body, many functions in our body. And the one, the nucleic acid that's responsible basically for coding this DNA. So for, I mean, not this DNA, for coding this protein, right, that we need. So protein, we use it in many things, in our hair, in our nails. Insulin is also made out of protein. There's a lot of functions for protein in our body. We need protein every day, and your body is even making protein right now as we speak, right? So protein is used in many different ways in the body. That's why we need to understand DNA and RNA, how it actually functions, right? And then, so okay, these are just the comparisons between the two. Okay, okay. Okay, these are the comparisons. Okay, we can move on. Okay, this is DNA replication. Like I said, when we're doing basically meiosis, right? Meiosis, okay, not meiosis, mitosis, right? So when you're doing mitosis, what will happen? We want to know how this thing replicates, right? Like I said, do you remember what I said? I said these bonds, these hydrogen bonds are what? Are weak, right? So they are weak hydrogen bonds, remember? So what happens is that, what will happen is that when this DNA, so it's ready to replicate, so it's ready for the replication. Firstly, you know that is a double helix, right? Helix means that it's sort of like it's something like this, right? So it goes on, and yeah, it's something like this, right? So what will happen is that it will firstly unwind, right? So double helix will unwind first, right? And then after it unwinds, it'll what? It'll unzip, right? So, and what does unzip mean? It just means that these hydrogen bonds will break, right? So these hydrogen bonds will break and then will unzip, something like this. And then what will happen is that now we have two sets, right? Two strands, right? You see two strands of so we have two strands of separate DNA, right? So I'll just call them half DNA, right? These strands, they will act now as templates, right? So do you see they served as templates? And what does it mean? It means that after this thing has unzipped, right? We have this part, right? This one part, this side, right? We have this one part, this side, and we have this other part, this side, right? Now, what did we say? We said these hydrogen bases are complementary, right? So now that this thing has unzipped, and now just a reminder, where did I say these nucleotides are found? They are found in the nucleus, right? That's why they are called nucleotides. So these nucleotides, all happen is that they are always free nucleotides, right? They are always free nucleotide in the nucleus. So all happens is that these free nucleotides that are around here, so these free nucleotides, right, will actually connect to each and every one of these. So this one here, these nucleotides will come and connect here. And like I said, we said what these hydrogen bases are complementary, right? So what will happen is that the ones that will attach here, do you see that this side, ne? So do you see that, let's say the left side, right? For this left strand, do you see that every nucleotide that will like basically bond to form this new DNA from this left side, the right side of it will be the same as the right side from the original structure, right? Because why? Because of these complementary bases, right? As you can see here, how it was in the beginning, right? A was connected to T, C, G, G, and all that happened is it unwinded and it broke, right? And then this side went this side. What happened is that when a free nucleotide comes, right, this T, you see, it was connected to this A. Another T is just gonna come and be slotted here, another G slotted here, C slotted here. Then do you see that this side, this left-hand side will have a separately new identical DNA structure, right? And then this side will happen. It will also act as a template. It will happen this side. There are three nucleotides. So A will connect here, C will connect here, and then also this one will connect another DNA structure that's also identical to this one. Do you see like what's happening here? Do you see? These two now are identical, right? Now it went from this one DNA to two DNA structures, right? So now that we have these two basically DNA structures, DNA replication is finished. So DNA, what is DNA replication? It's a process where the DNA makes an exact or identical copy of itself. Just like meiosis is a process where a cell makes an exact copy of itself, right? So the first thing that starts is this DNA replication, right? Right? And then, like I said, DNA replication is important for cell division, particularly mitosis, right? Because it allows chromosomes to be copied and to be sent to the daughter cells, right? Okay. So okay, this is just okay. They might assess it, Mare, maybe 2-3 marks, but some errors can occur during DNA replication, right? And when these errors occur, they lead to something called a mutation. So a mutation is basically a change in the structure, right? The structure of the DNA, the gene, right? So what will happen is that a new gene will be formed, right? A new gene will be formed because basically the incorrect nitrogenous bases will basically pair, right? And then a new gene will be formed, right? This may be harmful or harmless. Honestly, it depends on that particular person. And then this is also DNA. Okay, so there's a thing called DNA profiling. So DNA profiling basically, they can, using these DNA patterns, right? They can see basically if you guys basically are related, right? So if you are related, there are gonna be some genes, right, that they're gonna be the same, right? Especially if it's your mother or your father. Basically, half of that structure will be hers, or half of the structure will be his. So there are gonna be sections where you guys are going to be perfect, right? However, for identical twins, like the DNA profile will be exactly the same, right? Exactly the same for identical twins, right? That's because, okay, I'll basically tell you what identical twins are. So during the, so after the implant, right? So you know that there are two types of twins, right? They are non-identical and identical. So this is just the reproduction, this is for reproduction. So during implantation, right? If your body, like in your body, there were two, I would say two. So you had two eggs, right? And these two eggs were both implanted, right? Then you're gonna have two separate, so not identical twins, but you're gonna have twins, right? But they're not gonna be identical because each of them had their own egg cell, right? So each of them had their own egg cell. Do you see that? That's why they're still unique, even though they are they are twins. However, identical, what happened is that it was only one egg cell and one sperm cell that connected, right, and formed the zygote. But what happened is that when this zygote now expanded and created, so via mitosis created like basically another cell, right? These cells, they separated and then they grew separate, right? So what will happen is that now you have two two cells of the same structure that are growing, right? So obviously they're going to look the same because they have the same information from the father and the mother. But I will explain this also in reproduction, right? So DNA, like I said, it comes from your mother and your father. That's why, like, um for instance, maybe you and your brother, the match will be very low, or like compared to maybe you and your mother. One. Yeah, because don't forget that for your brother, even though the same mother, for your brother, there was a different egg cell that was used and a different sperm cell that was used, right? And during like meiosis, they talk about random arrangement and random selection and all of that. And all of those random arrangement and selections, they caused the egg cell and the sperm cells to not be the same. They are not the same, these sperm cells and egg cells, right? That's why, like, your brothers and sisters, like, you are further away from them than your mother and father when it comes to this DNA. So okay, this DNA profiling, obviously it's used for many things. I mean, it's used for identifying um suspects for a crime, right? It's because of this thing that each person has their unique DNA profile, right? So if they have your profile, they can just search it with what they found in the crime scene and determine whether you are there or not, right? And then, yeah, there are also many uses for this DNA profiling. I'm not gonna go through all of them. I just hope I will send you this textbook, right? But I think you do have textbooks, so I'll send you this textbook and then just go through all of these points. I want just read through them. You don't need to really cram them, just know them. Yeah, I'm sure they're gonna ask you maybe maximum, like I said, 3 to 5 marks this DNA profiling. And then now we are going to go to the reason why really we we are doing DNA and RNA. So the reason why we are doing DNA and RNA, like I said, it's protein synthesis, because we know that we have hair, we have nails. Like, how does the body do all of this, right? What instructs it? So like I said, the instruction is DNA. However, I will explain. Okay, let's start. So we want to know firstly how these proteins are made, right? So protein synthesis is basically how processing which proteins are made, right? So to synthesize means to make, right? Yeah. So to synthesize means to make. So when they say something is synthetic, it means that it's made, like it's obviously it's man-made. If like most probably when they say synthetic, they mean it's man-made. It means that it's not natural, right? In most cases, but the word synthesis comes from make. So a process in which proteins are made. So it's protein synthesis. And everywhere you see the synthesis, just know it means making it. And then what are proteins? So proteins are made out of what? Are made out of building blocks, right? So are made out of building blocks, and these building blocks are called what? I call these amino acids, right? And there are 20 different types of amino acids, right? And the way they combine, so the way they combine their arrangements and the length of the arrangements will determine what type of DNA is created, right? So do you see that because there are 20 amino acids, there are many different types of DNA you can, I mean, many types of protein you can create with this, because the variation can change. I mean, you can have 50 amino acids, right? Right, you can have 50 amino acids, each arranged differently, right? Obviously, if you have another 50 that's arranged differently from that 50, that protein will not be the same as that other protein, right? So the arrangement, so the sequence and the number of protein, the amino acids determine the protein that's being formed. So do you see that many proteins can be formed? So there's possibility of many proteins being formed, right? And then what will happen is that they are formed like this, or something like this. This illustrates the bond. So these are the different amino acids, and then they are bonded with this bond called a peptide bond. Like I told you, in life science or in these textbooks, they will show you which bond it's important to know. So the reason why they tell us what type of bond it is because they want us to know that this is a peptide bond. So in a test, they can ask you what type of bond is this. You have to know it's a peptide bond, and even the bond between those two nitrogen spaces, you have to know that those are weak hydrogen bonds, right? And then so right, so you see the smallest protein contains 50 amino acids linked together. Proteins generally contain 300 or more amino acids. Do you see? So there can be many, and I mean many different kinds of proteins that can be formed, right? And then, okay, so okay, I don't want to explain this first. Let's go here. Protein synthesis. So protein synthesis occurs in two stages. The first stage is transcription. The second stage is translation, right? And you'll see these stages, like these names, like they were put there for a reason. They mean exactly that. So what what does to transcribe mean? So what does to transcribe mean? Like I told you, DNA is there to send information, right? So this information has to be sent. So transcription is like the writing of the information, the sending of the information, right? So obviously, if let's say you're a boss, you have to write down maybe a letter to your to your um employees, right? Or some sort of notice, right? But you have to transcribe it down there. You have to relay it to them. You have to give the information. So the first one is transcription, where basically DNA takes this information it has and transcribes it. So transcribe means to transfer, transcribes that information to what? To the RNA. That's what transcription means. So I'll say it again. So firstly, transcription is where DNA transcribes, so passes its information to the RNA, right? That's the first stage, transcription, right? And I'll show you how this happens. So what will happen is, you know that the DNA firstly is made out of what? Weak hydrogen bonds, right? So firstly, these weak hydrogen bonds, so a piece. So okay, before we go further, DNA strands are long. We have these long DNA strands, right? These double helix strands, right? And in these in these double helix strands, all that happen is that sections of these so sections of this DNA. So let's say maybe this section here or this section here, right? There are sections to it, like a piece of this section that codes for certain information. Like here, do you see that there's a section that opened up? This section, it codes for specific information. So like I said, this DNA has a lot of information basically coiled into it, right? And then different parts of it, they are there for different sets of genes, so of information, right? Now some information is there are basically three types of genes, right? And there are genes, number one, that so they are non-coding genes, right? So like I said, a gene is only a six. So a gene, like I said, it's just a section of DNA that codes for something. So I can just say this is a gene because the section of DNA, but I'm not sure if it codes for something, right? So this is a six. So a gene is just a section of DNA that codes for something. So the first type of gene, like I said, it's a coding gene. So normal gene like this one, This is a gene because I'll tell you what, like the relation with this RNA right now, but this is a gene because it codes for information. And then the second type of gene is a section of DNA, right? That doesn't code for for DNA. That's called a non-coding gene. So that first one is a coding gene, and the second one is a non-coding gene. Right? So those genes, they do not code, they don't have like really information. And then there's a third type, and third and last type. These ones are just sleeping genes. Um, it doesn't really have much of a terminology, but these genes they are not active. Yeah, inactive genes. Yeah, so they are not active yet. Like for instance, the coding genes are active, right? They're coding and they're also active, right? Active means that the physical characteristics, like they show you, they can show, right, that you have this gene. And then asleep means basically, maybe let's say that gene you transfer it to maybe your child or something. Maybe let's say that gene was asleep in yours, like like when it came to you, right? Like maybe the genes from your fathers to you, it was asleep. Maybe when you transfer it to your child, that gene is going to be awake, right? And then your child is gonna have a characteristics maybe that your father had, however you didn't have, right? So it explains that. Understand? So there are three types of genes. So the non-coding, they don't do anything, those ones, right? And then we have the inactive ones, like I said, your father can pass you a gene which doesn't show on you, you pass it to your child has a gene that maybe your father has, right? And then we have active genes, those ones they show. So maybe, yeah, you have the same eyes maybe as your mother or whatever, so they already show, right? So they are active genes, those ones, right? And then what will happen is during this transcription, right, that part, so um the active obviously ones, right, the active genes, the coding genes, what will happen is that it'll that section will what firstly unwind, right? Just like here in protein in DNA replication. However, that unwinding will only be here for that section of the gene, right, responsible for that code. So what will happen then, it'll open, so these bonds will break, right? And then all happen is that there's gonna come this RNA, right? So you know that there are nucleotides right here, right? Like I said, there are nucleotides everywhere, and these nucleotides, like, they're different. Um there are different nucleotides with different bases, right? And like I said, now when it opens like this, it's not opening, so it can open for two reasons. Number one, it can open for DNA replication or for transcription. So when only a section of it opens, it's opening for transcription. But when all of it like opens up, then it's opening up for replication, right? So only a section here opened up, and then these free basically nucleotides, right? These free nucleotides, however, nucleotides relating to the RNA, right? So the ones that have the uracil base, right? What will happen is that it'll use part of this DNA, so this section of it, it'll use it basically as a template, right? So what will happen is that this mRNA will come and transcribe here, right? So this mRNA, this section will open here, and then these nucleotides will come here and form this mRNA, right? So we'll form this mRNA, right? So then this thing will form this mRNA, right? So after this mRNA is formed by that thing, this mRNA will go out, right? Will go out of this section, and then this thing will close up again, right? Now we have this mRNA. Do you see that this mRNA is built using information from this section here? Without this section, you wouldn't have this mRNA. Without this DNA opening, you wouldn't have this RNA, because this RNA is arranged, right, in the same arrangement as this DNA. Why? Because of these complementary bases. That's why I say these complementary bases are very important. They are the ones that the DNA uses basically to send out information, because different arrangements of these complementary bases, they give you a certain set of information, right? And all happen is that this mRNA, right? What is mRNA? mRNA is messenger RNA. Like I said, it's this information now it's transcribed. You see there, and then now we have this mRNA, this messenger RNA. Because it's a messenger, what does a messenger do? It transports the message. It has to transport the message. So it has to transport the message out of this nucleus to the cytoplasm, right? So let's go to the structure of a cell, right? So sorry, so structure of the cell is here. So it's going to go out of this nucleus and into the cytoplasm, so into the cell, right? It's going to go out of the nucleus via those pores and into basically the cells, right? And then what did we say? Okay, do you see? Zips, and then next as a template, and then using the free nucleotides in the nucleoplasm, right? Yeah, like I said, that's transcription. Do you see that this one is transcribing information to this one? Now we have this mRNA, and it was just transport. So it transports this information out of the cytoplasm into, so now it's into the cytoplasm. I mean, out of the nucleoplasm into the cytoplasm. So now we're going to the next phase of the protein synthesis, which is translation. Why? Because this information has been now transcribed to this messenger RNA. Now this messenger RNA has to translate the information. So what does translate mean? It means that you also give away the information. So transfer the information, right? Right. So translate, it means also transfer. So I'm also doing accounting. So let's say yeah, during maths, I get like maths, we also do foreign currency, I get. So let's say the rand is so every 1 rand is equal to $15. Ne? Let's say I tell you that write 3 rand in dollars, right? You're going to say it's 45. Now, if I gave you $3, right, and I say write those dollars into rand, right? You're gonna say it's 45 rand, right? But that wording is not really, it's not really standard English, right? What you had to say is translate, right? Translate the dollar amount to the rand amount, right? So now that's what translation means. So you are taking this information that you've transcribed, now you are also transferring it again. So these processes, they mean the same thing. However, you have to look at it, it's from the perspective of the RNA, right? It's from the perspective of the RNAs. It's transcription to what? To the RNA. The RNAs, the DNA is transcribing to the RNA, right? And then the RNA is what? Is translating. You'll see the RNA will translate, will transfer the information. However, you'll see that the information, it comes firstly from who? The DNA. So go to the second stage of protein synthesis. So this second stage occurs, obviously, they said it occurs in the cytoplasm, right? So what happens is we have the transfer RNA in the cytoplasm, right? So now, yeah, before we go on, so this thing, this mRNA, right? It's in the cytoplasm, and the cytoplasm basically in these ribosomes, right? So you've seen the cytoplasm has these small things called ribosomes. So it's in those things, the ribosomes, right? And then what happened is the tRNA, right? The tRNA, right? The tRNA. So even with like amino acids, there are amino acids inside like your cells, right? Amino acids, right? And then these amino acids, they attach to these tRNA, right? So these amino acids, they attach to this tRNA. And what is a tRNA, right? So a transfer RNA, well, what is it, right? So so now, like I told, there are many different types of amino acids, right? And even with these tRNA, there are different types of tRNA, right? But what is this tRNA? This tRNA basically is a nucleic acid, because obviously RNA is a nucleic acid that is used to transport an amino acid to a ribosome for what? For protein synthesis. That's what the tRNA is for. So it's for taking this amino acid, right? And then bringing it here for protein synthesis. That's its job, right? Now, how does it bring it here, and how does it now form this protein, right? So all tRNAs have this thing. So they have this part. This part is called the codon, right? And what is the codon? So basically, codon is we have basically three. You see, do you see three nitrogenous bases, right? These three nitrogenous bases are called codons, right? Because they are the ones that are actually help like with the coding of this amino acid. So codons come from the word coding. So these ones, they actually help with the coding, right? Right. So the mRNA will transfer this code it received from the DNA to these now amino acids through this tRNA, right? So what will happen is that there are different kinds of codons, right? So codons can like there are many also different types of codons, right? So because there are many different types of codons, there are many different types of tRNA, right? However, each tRNA is going to collect, so a certain tRNA collects a certain amino acid, right? So only a certain type of tRNA with obviously a certain type of tRNA. Obviously, tRNA is determined by what? By the codon, right? So do you see that a certain type of codon will take a certain type of amino acid, right? So it will be that way. So maybe codon with maybe I would say something like maybe adenine, maybe it has um, guanine and what's the last one? So it has adenine, it has guanine, and it has cytosine, right? So let's say a codon has that. That codon will have a type of amino acid here on top of it, right? So it will have a type of amino acid, right? So these tRNAs are just floating around, right? They are just floating around with these amino acids and these. So they are just floating around with these amino acids, right? Now, what will happen is that after the tRNA gets into this cytoplasm, right? These ribosomes, right? So during now this translation phase, what will happen is that this mRNA has a code, right? It has a code, right? What will happen is that this tRNA, they will come and they will join with these other hydrogen bonds. So these three hydrogen bonds will come here and join here, and this one with also complementary bases will join, and then these tRNAs will join, and as they join, yeah, so hey, the thing is, it's really difficult to illustrate here, but I'm gonna have to do this. So let's say this is the mRNA, so it has the complementary bases here, right? So these are the complementary bases. So what's going to happen is that, okay, now it's going to form bonds with this one, right? Bond with this one, bond with that one, right? The structure is very difficult to draw, but I'll try my best. So yeah, so that's basically the tRNA, and then we've got what? We've got our amino acid here, right? We've got our amino acid here, and then all happen is that another one will come, right? Another one will come, and then as this one comes, so as the next one comes, so as the next one comes, what will happen is that I get this one is here. So it'll come, okay, it'll join, it'll also join like this, right? Join, and then yeah, this is the tRNA. Then on top we have this amino acid, right? Now what will happen after this amino acid is here, right? This thing and this thing is going to bond via what? This thing called a, what did I say? I said this is a peptide bond, right? So they are gonna join via this peptide bonds, and what will happen is that the tRNA, this part here, it will disappear, right? It won't disappear, it will go away, I mean. So this tRNA will go away, it will go away and collect what? An amino acid that's similar to this and just float around again. So these tRNAs are just there for that, for taking an amino acid, floating around, and then if an mRNA comes, right, that has the same codon, it will go and insert the amino acid there, right? And then the same process will happen. Another one will come, tRNA, and as it comes, it will bond to this one and then form will form another bond like this, and then we have another amino acid here. Then these amino acid will arrange into this line, right? Like here. And you know that if you have 50 or more, then this is going to be protein. So that's how the protein is synthesized. That's how the protein is created, right? So now that's your protein. That's how it's created. That's basically protein synthesis in a nutshell. So that's what will happen. That's what will happen. So let's just read here what it says, right? mRNA codon, right? Oh yeah, sorry. The tRNA, this one is called an anticodon. Ne? Yeah, this is an anticodon. So tRNA has anticodons, and in mRNA, this three. So every three is called a codon, right? Yeah. And then obviously anticodon and codon will bond, right? Will join together, right? And then you see they are linked by a peptide bond to form protein. And this is basically showing the whole process. And then, yeah. So, okay, I did talk about mutation. And I think, let's check, just want to check. Okay, on the textbook side, okay, that's it. That's it. So that's basically protein synthesis, yeah. So like I said, there's two processes, transcription and translation. And you see that the most important things here are the mRNA and the tRNA. Honestly, to this day, I don't know what the other RNA is for, like the ribosomal RNA. I'll show you what I mean. So for now, don't even think about it because we don't know what the thing means. So there are the this one, ribosomal RNA, right? Oh yeah. So I really we know the function. So they say this ribosomal RNA, it forms ribosomes, right? Which are the site for protein synthesis. Like I said, yeah, protein synthesis, they occur, so protein synthesis happens in the ribosomes, right? I said ribosomes that are found in the cytoplasm, right? So these ribosomes here, right? So these ribosomal RNA, they create those ribosomes where the protein synthesis is, so where protein synthesis takes place, right? And then yeah, that's it. If you have any questions, please pass them via WhatsApp. And yeah, thanks.